A section C, or a long question, and how to approach it and how to break it down. So what you should notice in the long questions is that they're broken into 3 parts. So you have A and B and C, and they're always kind of broken up into 9 and 27 and 24. And generally speaking, okay, the marks are given in allocations of 3 marks. So if we look here at part A, it says, in relation to microorganisms such as bacteria and fungi, describe their distribution in nature. And what they're asking you here is, are you aware that they are basically found everywhere in most places all around the world? And that answer will be worth 3. And then, you can kind of see here, that it says, explain the terms asepsis and sterility as applied to microorganisms. And they are italicized, so they're asking you to define these words, and each of those would be worth 3 marks. Okay, so the next thing that we need to move on to then is drawing diagrams. And what I'm going to do here is I'm going to take this question. So the question says, draw the structure of a bacterial cell and label any 3 parts. So what I've done here is I've taken a page from the Leaving Cert section C answer book, and I've put it in here into OneNote. And I'm on part B, I. So let's just say this was question 12. And it does always say start each question on a new page, just to be clear on that. I'm now working on part B, and I'm working on the first part. And the question is, draw a bacterial cell and fully label it. So again, you cannot get any marks for a diagram unless it is labeled. So I'm just going to really quickly here sketch a bacterium. This could be a rod-shaped bacterium called a bacillus. You need to remember that bacteria do not have a nucleus, so I'm just going to draw the piece of DNA here like that. I'm going to draw in a plasmid here. I'm going to put in a couple of ribosomes, and because it's a bacterial cell, I might just put in a couple of little flagella like that. So at the moment, just so you're really clear on this, this is getting 0 marks. And the reason it's getting 0 marks is there's no label. So at the moment it's D0 and it's L000. So the way this question is marked, it's 6 marks for the diagram, made up of 3 for the diagram itself and 3 by 1 for the labels. If I even do this, and I put in a labeling line and I write down cell wall, now this student moves from getting 0 out of 6 to getting 3 for the diagram and 1 for the label. And if I just want to box this off and make sure I get all the marks, I can now add in a couple of more labels. I can say that that's a plasmid, which is a circular extra piece of DNA. I can say that that is a ribosome. You need to remember that bacteria have ribosomes because they need to make their own proteins. That could be the cytoplasm. This could be the DNA. And I would say to try and put in as many labels as possible. That is the flagellum, or the tail. Bacteria use flagella for motility or for moving. And now this student is up to the max, which is 6 out of 6. So if we go back up and look at the rest of the question and just try to break it down a little bit more. So a phrase that I want students to be aware of is the phrase command words. So when you're looking at the question, you need to be really, really conscious of what the command words are. So in the first example there, it was the word draw. And then the next question says, are bacteria prokaryotic or eukaryotic? And then this phrase that has come through from junior cycle science, justify your answer, means, are bacteria prokaryotic or eukaryotic? You give your answer, which would be prokaryotic, and then justify your answer basically means give a reason. Bacteria reproduce asexually. Name and describe this process. So the command word here, the first one, name, write one thing. So the answer is binary fission. Then the next command word or phrase is describe. So here you need to write in a number of bullet points. So binary fission, you'd probably be looking at 3 to 4 bullet points and a couple of diagrams. So just get used to saying to yourself, okay, what are the command words? What am I being asked exactly in this question? And then the next one is in italics. So it's a definition. What does pathogenic mean? Disease causing. And then antibiotics are used to treat pathogenic bacterial infections, but they're often misused. Give a possible effect of the misuse of antibiotics. And the answer here would be that they build up resistance and that eventually some antibiotics can become useless. So it is important that you're able to break down the 24 marks here or the 27 marks here and work out how much to write. So if I do a version of this here, it says, I'm going to skip the first part because it says the command word explain. So when I'm breaking these down with my students in class, I always go for the easy command words first and then work away at trying to work out how many marks are going for the longer pieces. So name the 2 biochemical components. Now, that is protein and DNA or RNA. So there'll be 3 marks for each of them. So the whole thing is worth 24, and we're up to 6 out of 24 so far. It then says, give 1 example of a harmful virus and 1 way in which viruses can be beneficial. There will be 3 marks for each of them. So now that's 12 out of 24. And then it says, explain the difficulty in describing viruses as living organisms. This could be a sentence. This could be, okay, viruses do not demonstrate all of the characteristics of living things. They cannot reproduce. They can only replicate. So that will usually only get 3 marks. And so now we're up to 15 out of 24. And what that means is that this part 3, because of that command word describe, is going to be worth 9 marks in total. And so you will have to write a minimum of 3 bullet points or combine 3 little bullet points with 3 diagrams to get the full marks here. And so that's how I would kind of start practicing when I'm doing my exam questions on my own, when I'm studying for my house exams, for my mocks, for the Leaving Cert itself. Start breaking these questions down. Be very conscious of the command words and be very conscious of all the other things that I've gone through in the webinar so far.